Welcome, this is Mrs. Kinlan, and in this video I would like to introduce you to our Daphnia magna experiment. The purpose of this experiment is to find out if alcohol acts as a stimulant and raises the heart rate of Daphnia magna, or whether it acts as a depressant and reduces the heart rate. So first of all, we're going to need a container of Daphnia magna. We have our pipette where we have already removed the tip to make a wider opening to capture our Daphnia. We have spring water that we will use as our control setup, and we will need to make a 3% alcohol solution. So we'll take our spring water into our graduated cylinder and fill it to 97 milliliters, and then we will add 3 milliliters of rubbing alcohol, and that is what makes up our 3% alcohol solution. So we also have to make sure that we use a depression slide. So make sure that you put the Daphnia on the correct side. The bottom should be smooth and flat, and on the top you should feel that little depression. So we will use our pipette that's cut, and we will capture a Daphnia, and we'll just put it in a couple drops of the nice spring water. And we're going to put it on our stage of our compound light microscope. Make sure that you lift up and that you use the stage clips. And then just center the Daphnia over the light area. To turn on our microscope, we're going to twist the dial. I barely turn it on so it doesn't make it too bright or too hot for the Daphnia. And then we want to start with the stage all the way up. So I'm going to use my coarse adjustment knob, and I'm going to raise the stage, and I'm going to look inside my eyepiece, and I'm going to use my fingertips to center the Daphnia in the center of the stage, and I'm going to slowly lower the stage and until the Daphnia comes into view. So once the Daphnia comes into view, I want to focus on the back where the shoulder blades would kind of be. This is where we're going to locate the heart on our Daphnia. You'll see a very large eye spot, and you'll see some appendages moving. You'll see like a brown digestive system moving throughout the Daphnia, but you want to look on the back side, and you will see a small, clear heartbeat, and that's what we want to focus on. So once we have, if the Daphnia is moving too much, what we'll want to do is take a few strands of cotton and lay this over the Daphnia or in the water, and the Daphnia will bump into the cotton and kind of stop moving out of your field of view so much, so that will slow them down. So once you have it so that you can see the heartbeat very easily, what you want to do is take a felt tip pen and a piece of paper, and your lab partner can keep time for you. You want to count the number of heartbeats in 10 seconds. So they will be your timer, and then they'll tell you, you can say you're ready, and they'll tell you when to go, and you just tap out the heartbeats for 10 seconds. When your lab partner tells you to stop, find a new area on your sheet and tell them you're ready and go, and this will be trial number 2. Tap out the heartbeats, and then they will tell you when to stop, and one more trial. So find a fresh spot on your paper, tap out the beats for 10 more seconds, and then what we want to do when it's time to stop is turn off your light bulb so it doesn't heat up the Daphnia artificially, and you want to count your dots. And if the numbers for the 3 trials are within plus or minus 10%, about 10% of each other, then that is good, and you want to keep that data and record it into your data table. If you have one trial where the number is way out of range, very different than the other numbers, then you want to right now go ahead and repeat that trial and do that. Once you have 3 trials of good data, then what we want to do is take our pipette and get a couple drops of the alcohol solution. So now we're going to do our experimental setup. We're going to find out how alcohol affects the heart rate. So we're going to add a couple drops of alcohol solution onto our Daphnia, and then we're going to let that penetrate the carapace, the exoskeleton. While that sits for about 5 minutes, now you can keep time for your lab partner. So you'll be the timekeeper, and they are going to do their control setup, 3 trials in spring water. Once they've counted their data and accepted it, your lab partner will put the 2 drops of alcohol solution on their Daphnia and let that set for 5 minutes. And during that time, now you're going to go back and do 3 trials of alcohol. And then be sure you count those numbers before you put your Daphnia, your finished Daphnia, in the used container when you're all done. Okay, so that is our Daphnia Magna investigation, and I hope you enjoy it. Thanks.